everyone, I am Romane Junker and I am currently doing the second year of my PhD in the maillage laboratory at the Paris-Saclay University. My PhD focuses on bacterial communities from lacto-fermented vegetables, and I will present my work on integrated metagenetic datasets through microbial association network to compare microbial community from lacto-fermented vegetables. So, the development of low-cost sequencing technology has generated a massive amount of microbiome datasets during the last 20 years. They represent an unprecedented opportunity to better understand microbial ecosystems. Nevertheless, it also raises many challenges. This work integrates public metabarcoding datasets related to lacto-fermented vegetables. There is currently renewed interest in the bacterial community of fermented vegetables, as plant-based fermented food are at the heart of sustainability and health issues. Lacto-fermented vegetables are the product of a relatively well-described spontaneous lactic acid fermentation by lactic acid bacteria. However, community succession has received little attention in lacto-fermented vegetables. This study presents an integrative bioinformatic approach to explore and compare public time series metabarcoding datasets on vegetable fermentation by building amplicon sequence variant (ASV) association networks. You can see here the developed workflow. So we start by collecting datasets from NCBI SRA, Magnify and Food Microbionet, and we selected those with several time points, more than 10 samples in the study, an associated publication to have access to metadata, and the same RNA region sequenced. The 10 selected datasets come from different vegetables, such as cucumber, carrots, pepper or cabbage, and contain between 11 and 206 samples and 32 unsynchronized time points, as you can see here. Then we applied the data to pipeline to those datasets to obtain an ASV count table for each experiment, and we use this table to infer a microbial association network for each experiment. We use 4 metrics to reflect co-presence and co-abundance. For instance, here you can see a microbial association network built from 18 samples during 6 time points. Here the samples are ordered by sampling times. And in the network, each ASV corresponds to a node whose size depends on its maximum related abundance, and color represents its taxonomic order. For instance, yellow is for Pseudomonales, violet for Enterobacterales, and blue for Lactobacillales. The x-axis corresponds to the weighted mean age, WMA, of the samples where the ASV is detected, measured in days and weighted by the relative abundance of the ASV. An edge is shown between 2 nodes if there is an association found according to at least one metric. The association network highlights the dynamic evolution of microbial community during Paukai fermentation. Here, the network is composed of 2 distinct patterns, a high diversity of ASV belonging to the initial vegetable microbiota, and on the other hand, Enterobacterales and a majority of Lactobacillales. Finally, we compare the 11 different networks obtained by building a core network with edges found in at least 3 networks, and clustering the ASV according to their connection pattern by an SBM clustering. Here you can see that the core network highlights that there is a shared temporal structure with a shift from the initial vegetable microbiota to Lactobacillales through Enterobacterales. Studying the ASV prevalence shows that the initial microbiota as well as the lactic acid bacteria present mainly at the end of fermentation are more specific to each experiment. And the clustering results are interesting since you can see that here cluster 5 in bright green highlight an association between Enterobacterales and several lactobacillides. So this community may represent an early fermentation core consortium. To conclude, the comparison of microbial ASV association network is valuable for exploring bacterial community successions and guiding the exploration of potential microbial consortia responsible for different fermentation phases in lacto-fermented vegetables. This work validates the recurring patterns of Enterobacterales presence at the beginning of the fermentation, thus raising questions about their ecological role. However, the biological interpretation of this work is limited by its scale and the metabarcoding technology. Ultimately, this study highlights the potential of reusing data to better understand microbial ecosystem and the need for complementary functional studies such as shotgun metagenomics or metatranscriptomic with sampling and monitoring over time to improve our understanding of vegetables fermentation. Thank you very much for your attention.